And I think we can go ahead and get started. So good afternoon, everyone. It's my pleasure to introduce my friend and today's seminar speaker, Dr. Sathish Srinivasan. Sathish is an associate professor in the Cardiovascular Biology Research Program at Oklahoma Medical Research Foundation. He started working on the lymphatic system as a postdoc in Guillermo Oliver's lab. And as a postdoc, he provided further mechanistic insight into the role of PROX1 in lymphatic development. And he also created the first tamoxifen-inducible PROX1 mouse, which has been widely used in the field. Sathish started his own lab in 2013 and has made seminal discoveries on the development of lymphovenous valves and lymphatic valves. And his lab continues to use genetic approaches to investigate the role several signaling pathways serve in the development of the lymphatic system. And he also studied the mechanisms by which lymphatic endothelial cells respond to shear stress. So the title of his presentation is Shear Stress Regulates Lymphatic Vascular Patterning. Welcome, Sathish. The seminar is yours.
Thank you, Mike. Thank you very much for this kind introduction and for inviting me to this seminar series. I am a basic investigator. I study the basic mechanisms of lymphatic vascular development, but I have always been wanting to find the connection between what we find with lymphatic malformations, which is of interest to your community. And I hope you'll be able to provide some insights, see something from a different angle, and give me some suggestions to explore. So I please keep it interactive. Please stop me whenever, if you have any questions or any suggestions, and we can go over it. So let me start with the very basic information about the lymphatics. So the lymphatic vasculature is a hierarchically arranged vascular tissue. So you have the capillaries. The lymphatic capillaries are the main functional units of the lymphatics. They have what are known as button-like junctions with a lot of gaps in between. And through these gaps, the interstitial fluid or immune cells, they are able to enter the vessels, the lymphatic capillaries. And there are these structures, I don't know whether to call them hypothetical or real. There are some electron microscopy images that seems to suggest that these structures known as anchorin filaments are critical for the opening and closing of lymphatic capillaries. But these are poorly understood, and there are very limited studies exploring the role of these structures. And from the capillaries, the fluid and the immune cells, they are transported into what are known as collecting lymphatic vessels. And here the cell-to-cell junctions are tighter. There are fewer gaps in between the endothelial cells, and that prevents the leakage of fluid back into the interstitial space. And there are lymphatic valves which regulate the flow of fluid within these collecting lymphatic vessels. And there are specialized cells known as mural cells, known as lymphatic muscle cells, which pulsate just like cardiac muscle cells that to propel the fluid forward. And these collecting vessels are there are lymph nodes which are located within the collecting vessels, and there the lymph fluid is filtered. So the immune cells within the lymph nodes can identify any potential pathogens like bacteria, or viruses, or fungi, and they can trigger the acquired immune response. And finally, the lymph fluid is returned to blood circulation via exactly four valves in the human body or in any mammals, known as lymphovenous valves. So this is a vein. This is a junction of internal jugular vein and subclavian vein, and the heart is located just underneath it. And it's basically taking advantage of the diastolic vacuum created during heart function there is a lot of vacuum created during heart pumping, and the lymph fluid, because it's very low pressure, it's taking advantage of this connection and it's getting drained back into the blood circulation. So lymphatic anomalies can be, people like Mike and others have worked hard to classify these lymphatic anomalies into several categories. And they can be of tumor types, where the lymphatic endothelial cells proliferate rapidly, or they can be lymphatic disorders such as lymphedema, or lymphatic anomalies that are also associated with other tissue defects, and I'll have some pictures to show you. And then there are lymphatic malformations. So here are some of the classic pictures, and here is a macrocystic lymphatic malformation, where you have this big cyst, very commonly seen in the neck area of kids. And these are, to my understanding, they are treated relatively easily with surgical methodologies. And these are microcystic lymphatic malformations, which are a little harder to treat because they are more dispersed, and their connection with the blood vessels are pretty hard to identify. And here are the lymphatic malformations that are associated with other disorders like the limb overgrowth and tissue overgrowth, bone overgrowth kind of defects, various disorders like the Close and Clappo syndrome, PWS, etc. And very commonly, so these are the pathway, these are mutations that are found to be associated with the lymphatic malformations. And the most commonly associated mutations are in the MAP kinase pathway, activating mutations in RAS molecule, or activating mutations in PI3 kinase, especially PIK3CA mutations are associated with the lymphatic malformations. Now, I am, as I mentioned, I don't work on these mutations. I don't study the consequences of activating mutations in RAS and PIK3CA. However, my interest is in the mechanisms which are upstream of it. And I'll have one particular, I should have mentioned this one. So these mutations, these activating mutations in RAS or PIK3CA, they appear to compromise the basic lymphatic architecture, right? So I mentioned to you about the capillaries, which have button-like junctions. They appear to, this architecture appears to be changed. They seem to have a lot of zipper-like junctions in the capillaries. And instead, there is a lot of leakage from the collecting lymphatic vessels. And the lymphatic valves, they appear to be compromised. They are either they are fewer in numbers or their structure is very defective. And I don't believe there are any studies about the lymphovenous valves or lymph nodes in this context, but I wouldn't be surprised if those are defective as well. I'm sure there are groups working on it. So as I mentioned, I don't necessarily work on activating mutations in KRAS or PIK3CA, but my interest is upstream of it. So what are the molecules that activate MAP kinase or PI3 kinase pathways, especially with respect to particular receptor tyrosine kinase known as VEGF-R3, which is considered the most upstream and very critical lymphangiogenesis signaling molecule. And we have also got an interest in a particular G protein coupled receptor called S1PR1, which is important for regulating these ERK and MAP kinase pathways. And also shear stress. The endothelial cells, like the lymphatic endothelial cells and blood endothelial cells, are constantly exposed to the flow of fluid, right? In our case, it's the lymph. And this flow of lymph can activate PI3 kinase and MAP kinase pathways. And that is another aspect that we are studying. Okay. So now to the basic biology, right? So when the lymphatic vessels are growing, this VEGF-C signaling, which it's the ligand that associates with VEGFR3, it's the most important lymphangiogenic cue. So the lymphatic endothelial cells are migrating towards the source of VEGF-C. At least this is the predicted model. And as these cells are growing towards VEGF-C with this philopodia and very highly active state, the cells which are following it, the cells which are just behind this actively migrating endothelial cells, have to acquire their characteristics. They have to become quiescent. They have to acquire the characteristics of capillaries or collecting vessels. The lymphatic valves need to form. They have to recruit the lymphatic muscle cells. So it's a very interesting aspect how on the one hand, the vessels are migrating and on, simultaneously they are also becoming more mature and quiescent and properly patterned. So this is the question that we wanted to address, right? And these are two models that we use. One is the dorsal skin model. So if this is a mouse embryo, and the lymphatic vessels are migrating from both the ends, right? So on the left side and right side, there are lymph sacs. And from the lymph sacs, the lymphatic vessels are migrating towards the midline of this dorsal skin. So by looking at the lymphatic vessels at various time points, we can get an understanding about how the lymphatic vessels are growing and undergoing maturation. So this is one model that we use. And the second model that we use is the mesentery in the gut. So this is the gut intestine of embryo. And the lymphatic vessels are growing from the center. So there is a lymph node here. And the lymphatic vessels come from the lymph nodes where the lymph sac, primitive lymph sac would have been. From that lymph sac, the lymphatic vessels are migrating along blood vessels towards the gut wall. And then it penetrates the gut wall and then it forms the lacteals inside the gut. So these are two models that we frequently use for studying the growth of lymphatic vessels. So using these models, we wanted to investigate the role of this G protein coupled receptor called S1PR1. And the reason we decided to investigate it is that it's already known in blood vessels that S1PR1 inhibits the growth of blood vessels, and it's critical for the patterning of blood vessels. So this is the retina of mouse embryos, retinal blood vessels of mouse embryos. In control mice, you can see the blood vessels which are nicely formed. Here you have arteries and veins that are in the middle, and then you have the capillaries that are nicely patterned in between, connecting the arteries and veins. But if you delete S1PR1, one thing you can notice is that the arteries and veins are defective. There are too many branches, abnormal branches, but the density of the capillaries is also increased. And if you look at higher resolution, higher magnification, you can notice a lot of philopodia, the tip cells in these mutants. So we asked a very simple question whether S1PR1 is playing a role in lymphangiogenesis as well, and if so, to understand the mechanism of action. So we started with this reporter. This is a very elegant reporter that was generated by a group in NIH. So the idea is that if S1P, this is the S1PR1 receptor, and when it engages with its ligand, S1P, it's a biolipid. When the ligand engages with this GPCR, it results in the activation of a GFP reporter. So it's basically a readout for the places where the reporter is active, the receptor is active. So we use that reporter to look at the lymphatic vessels. This is again the dorsal skin of the mouse embryo. And GFP is in green here, and the VEGFR3, that's the lymphatic vessel, in red. And you will notice that there is this beautiful, very interesting pattern of GFP expression. So GFP, indicating S1PR1 activity, is kind of weak in the migrating tip cells. So the vessels which are growing fast are devoid of S1PR1 activity, but the vessels which are more mature, they are becoming quiescent. They are not proliferating or branching so much. They have very strong GFP expression. And also, GFP is also expressed in the developing lymphatic valve of this embryo. So it gave us a hint. It suggested that S1PR1 could be important for the quiescence and maturation of lymphatic vessel. And what I'm showing here is a control embryo where there is no reporter activity as a negative control. Okay. So with that background, we deleted S1PR1. We took this Cre line known as live Cre, which is active, at least in the skin, it is more or less specific for the lymphatic vessels. We deleted S1PR1 using this Cre line, and you can notice two things happening here. So here in a control embryo, the lymphatic vessels have reached the midline. This is a stage called 16, 17 and a half. The lymphatic vessels have reached the midline, and the vessels are very smooth and they are very nicely patterned. So there are gaps in which, so there is sufficient amount of interstitial space in between the lymphatic vessel where the fluid can be absorbed by the lymphatic vessel. Whereas in this particular mutant, the vessels have not reached the midline, but instead the lymphatic vascular density is dramatically increased. There are more branches and the diameter of the lymphatic vessels are substantially increased in these mutant embryos, which we have quantified here. And additionally, if you look at these lymphatic vessels at very high magnification, higher magnification, you can notice that in control embryos, the surface in the more mature vessels, the vessels have a very smooth surface and without any tips or sprouts. But instead, when we delete S1PR1, you can notice all these filopodia and tip cells that are coming from places where they normally wouldn't have formed. I hope it's obvious for you. And as I mentioned to you, VEGF-R3 signaling is the most potent or most critical lymphangiogenic signaling pathway known. So we tested whether S1PR1 could be antagonizing this particular signaling pathway. And the way we did it is by removing one allele of VEGF-R3 in the context of S1PR1 mutant. So we deleted S1PR1. And again, you can hopefully see the tip cells and filopodia that are abnormally coming from the lymphatic vessels. And when we removed one allele of VEGF-R3, those filopodia and tip cells were substantially reduced. Significantly, we were able to rescue the phenotype. So this observation indicated to us that S1PR1 is inhibiting VEGF-C, VEGF-R3 signaling cascade. So we started to study how it could be doing it. So one of the basic things that we do in the lymphangiogenic or angiogenesis pathway field is to treat primary human endothelial cells, either lymphatic or blood endothelial cells, with the morphogen, that is either VEGF-A or in this context it is VEGF-C. So we treated the cells with VEGF-C either in a control context or in cells in which we knock down S1PR1. As you can notice, S1PR1 levels are reduced in these cells, and we treated those cells with VEGF-C, and it activates the molecules that you are very interested in, ERK and AKT. They are activated by VEGF-C. But when we knock down S1PR1, we didn't see any obvious reduction or no obvious change. They looked comparable in this particular, and that was very surprising to us. So next, we looked into the shear stress response. So as I mentioned to you, S1PR1 activity is more is stronger in these mature lymphatic vessels, which are exposed to shear stress. The fluid is flowing, the lymph is flowing, and that is where S1PR1 is active. In contrast, normally, VEGF-C will be more active. VEGF-C activity will be more active in this tip cell area. So that made sense. So we decided to look at the role of S1PR1 in regulating shear stress response. And people have done quite a bit of work in what is known as the mechanotransduction pathway, right? So one thing that happens is when you grow culture cells in the presence of shear stress, in this particular case with laminar shear stress, the cells align, they become thinner, they become elongated, and they align along the direction of either blood flow or in this case, lymph flow. And when we knock down S1PR1, we didn't see any obvious changes. The cells seem to be aligning just the same. They look normal. And the other thing that happens is that when you grow endothelial cells in the presence of shear stress, in this case, laminar shear for 10 minutes, once again, you can notice that the cells are activating AKT and ERK. This is very well known, and it also activates other signaling cascade like P38 and ENOS. These are all activated very quickly, very rapidly, it's increased. And when we inactivate or downregulate S1PR1 expression, we don't see any obvious reduction or increase, any change in any of the signaling cascades. So again, this is once again very surprising. So it doesn't affect VEGF-C signaling, and it doesn't affect shear stress signaling. And finally, we also tried we did an RNA-seq experiment to identify molecules that are regulated by S1PR1. So we grew cells in the presence of laminar shear. And one very interesting thing that happens is that it downregulates the expression of various molecules like DLL4, adrenomedulin, ESM1, CD38, angiopoietin. And what are these? These molecules are tip cell markers. So I showed you that nice figure where you have the tip cells and you have the mature vessels. And these cells are expressed in the tip cells, and consistent with the idea that the shear stress downregulates or makes the vessel more quiescent. These tip cell markers are downregulated by shear stress. But once again, when we do this experiment in the presence of siRNA for S1PR1, we didn't see any obvious differences. So laminar shear is able to still downregulate the expression of these tip cell markers. So, we were frustrated at this time, but we came up with this hypothesis, and I don't know how we came up with this hypothesis. We decided maybe it's a combination of both. So it is not just S1PR1 is inhibiting VEGF-C signaling, or it's inhibiting or promoting laminar shear, but maybe it's a combination. So maybe laminar shear is regulating VEGF-C signaling, and then S1PR1 is playing a functional role, a critical role in regulating this shear stress induced or regulated VEGF-C signaling pathway. And that's what we tested, okay? So we grew the cells either under static conditions, that is without any shear stress, or in the presence of laminar shear for 24 hours. And then after exposing those cells, culturing those cells like that for 24 hours, we treated them with the VEGF-C for 10 minutes, right? It's a quick exposure. And what you will notice, this is all in control cells without any knockdown of S1PR1. And you will notice, compared to cells that were grown in static condition, the cells that were grown under laminar shear had very high phospho, ERK, and AKT activity. And this could be very relevant for your studies, the lymphatic malformation context, whether this could be relevant, you may want to think about. So, so the first point is that in the presence of laminar shear, the expression VEGF-C is able to activate ERK and AKT very potently. There is this very potent activation of these pathways. And then when we repeated the experiment, now in the presence of S1PR1. Now, all the cells were all grown in the presence of laminar shear, and then we downregulated S1PR1. And I hope you can see, this is a very subtle difference, but you can hopefully see that compared to control cells, when we knock down S1PR1, we are able to see an enhancement in the expression, especially of ERK, more so than AKT. There is a mild increase in AKT, but ERK is more prominent than AKT. And you can, once again, you can see that there is this increase in phosphorylation of ERK status. And we also look for certain genes which are known to be responsive to VEGF-C signaling. And one of these molecules is known as EGR1, that is early growth response molecule. And this is known to be regulated by many receptor tyrosine kinase signaling pathways. So here again, we cultured cells either statically or in the presence of laminar shear. And then we treated with the VEGF-C. And you can notice when we treat cells just with VEGF-C, it enhances the expression of EGR1. This is after 1 hour of treatment. And when we express the cell, when we grow the cells in the presence of laminar shear, and then treat with VEGF-C, we see nearly a 20-fold increase in the expression of EGR1. That again confirms that laminar shear is able to enhance VEGF-C signaling. And then when we knock down S1PR1 and do this experiment, there is at least a 2-fold increase again in the expression of EGR1, again confirming that the absence of S1PR1 is able to enhance MAP kinase pathway or a VEGF-C signaling pathway in those cells. Okay. So this is the conclusion for our study, right? So in a control context, you have laminar shear that does 2 things. Paradoxically, it's doing 2 things simultaneously. One, it inhibits the expression of tip cell markers, which are important for sprouting, of course. And on the other hand, it's enhancing VEGF-C signaling, which could do 2 things. It can promote sprouting, but in the presence of S1PR1, it's able to regulate the quiescence and maturation of lymphatic endothelials. And I can go through this a little more detail because ERK, MAP kinase pathway, it's known to be very important for quiescent endothelial cells. So if you delete ERK1 and ERK2 from endothelial cells, the vessels, the blood vessels will just completely disrupt. Even in adult mice, if you delete ERK1 and 2, these mice will die in no time. Within a week, they are dead. So ERK, the MAP kinase pathway, is very critical for maintaining the quiescence of endothelial cells. And S1PR1 is basically playing this role in shunting the MAP kinase pathway to maintain the quiescence of endothelial cells. But on the other hand, if you don't have S1PR1, then laminar shear is enhancing this MAP kinase pathway, and that is promoting, causing the hypersprouting of endothelial cells, specifically in our context, it's the lymphatic endothelial cells. And we can, once again, we can model it here. So in the tip cells, we don't have S1PR1. So these cells are growing towards a very high VEGF-C gradient. They are sprouting, they are migrating. And the vessels which are quiescent, the vessels which are exposed to laminar shear, that is where S1PR1 is also active. And these are places where ERK activities, there is this balanced ERK activity that maintains the quiescence of endothelial cells. And in places where the vessels, either lymphatic vessels or blood vessel that are not connected to blood circulation or lymphatic circulation, this you would expect them to be inefficient. They are useless vessels, which are not playing any functional role in draining lymph fluid. So here, the VEGF-C signaling is going to be defective. It's so that the flow, shear flow, is not present to sufficiently activate VEGF-C signaling. So I would predict that these vessels will regress. They will disappear. Therefore, only the most useful, the most efficient lymphatic vessels will remain in order to transport the blood fluid or the lymphatic fluid in our case. So this is the basic biology, right? So how S1PR1 could be relevant in lymphatic malformation, for example, will be very important to study. So there are molecules that are known to activate S1PR1 signaling. So can we utilize those molecules to somehow cause a regression of, I would predict that this is what is happening in lymphatic malformation. You have certain areas of lymphatic vessels which are ineffective. They are not transporting fluid, but they are growing and they are just keep accumulating fluid inside. So will it be possible to promote the regression of those structures by using S1PR1 agonist? Will be something that you can consider. Okay, so that is the closest I have, with the lymphatic malformation. But here, this is a paper that we published a couple of years back, and we found this observation which was really excited me, and I contacted Mike to get his suggestion and thoughts. So we deleted YAP and TAS in lymphatic endothelial cells using this particular Cre line called PROX1 Cre. And the catch with this Cre is that it's also heterozygous for PROX1. So when we delete one allele of PROX1, one allele of YAP, and one allele of TAS, you can see that there are some defects here. You can already see that the lymphatic density is somehow increased, but the vessels are not migrating close enough. But then when we push it even further, when we delete two alleles of YAP and one allele of TAS, then we start seeing these kind of structures with which are, I would call it microcystic, but I don't know. But these mice die. They don't survive very well, and so we may have to investigate by different approaches in postnatal mice, and hopefully we can get it there. But whether this PROX1 and YAP and TAS, whether they have any role in MAP kinase and PI3 kinase pathways, will be something to explore. But if you have any suggestions, if it reminds you of something, please let me know. Well, finally, I would like to thank the people who have done this work. And this S1PR1 project was done more or less exclusively by Shin. She is my wife, and she just left the lab. We have been working together for the past 10 years, and she has been very instrumental to get us until this stage. And she decided to be independent and try something new. So she has joined a startup company in Boston just this month, and hopefully everything will go well. We will see. And Yenchun and Li Juan, they have been in part of the S1PR1 project as well, but they are doing other things. Zoheb, Zayla, and Stephanie, they are newer people in the lab. They just joined in the past few months, and they have picked up a few ideas from this work. And collaborators on this project have been Dr. Tim La from Harvard and his postdoc, Keizu, Hong Chen, they have been very instrumental. Then Yang Kwon and Dong Won from USC, they have helped us establish the shear stress model, and they have generously shared a lot of cells with us. And then Konstantinos, when he was at Texas Tech, helped us with a study that I didn't present here. We explored the role of S1PR1 in vascular stabilization and GPCR pathway. And Joshua also, he worked with us on this project. I didn't show the data about the role of DLL4 in signaling. And Dr. Hirotaki Chisi from Tokyo, he gave us the VEGF-R3 mouse model. So with that, I will stop and I will be happy to take any questions that you may have. And thank you again for spending 40 minutes or 1 hour with me. That's very generous of you. Thank you.
Thank you, Sathish. That was a beautiful presentation, really exciting work. And I'll get the discussion started. So I was curious if there's, if you looked to see if there was a relationship between S1PR1 expression and proliferation markers. So are they like mutually exclusive in terms of their expression?
Actually, we did Ki67 staining. We didn't exactly see a dramatic change in proliferation. And the way I look at it is the proliferation, the most significant, at least when I did this study long back, the most significant proliferation happens when the lymphatic endothelial cells are coming out of the vein and forming the lymph sac. So at that stage, you can see a rapid PH3, phosphohistone 3, and also Ki67, a very rapid proliferation. But once the lymph sac is formed, and once those endothelial cells are sprouting from the lymph sac towards the midline or towards the organs like lungs or heart, it seems to be happening by, so the lymph sac size becomes smaller and smaller as the vessels sprout and sprout. So it seems like it's, I don't know what's the correct term, or I'm missing it. So the lymph sac is kind of squishing in and squeezing out to form those lymphatic vessels without any significant proliferation happening.
And
so
I just wanted
to address your question.
Yes, and I just wanted to highlight, I mean, how important I think this concept of quiescence is to vascular anomalies in general, either this loss of quiescence or a failure in becoming quiescent, I think is a really interesting concept for vascular malformations. And I think it would be really cool to stain for S1PR1, some of these vascular malformations, to see, you know, do we see is there a relationship with that and this loss of quiescence?
Yeah, that's a very nice idea, Mike. I think that has to be done. So this reporter is available from Jackson Laboratory. And you can, you know, you have worked with the KRAS mutation and PIK3CA mutation. So, you know, they can be bred together, and then we can exactly pinpoint what kind of defects there could be in the S1PR1 pathway. Yeah, that's a very good idea.
Great, and we'll keep the discussion open. So if you have a question, please feel free to unmute yourself. Or alternatively, you can type your question in the chat box and I'll read it.
Not many questions today.
Yeah, it was a very clear presentation, and it was, I think people are excited about this interesting concept and maybe need more time to think about how they can relate it to what they're seeing.
Yeah, please don't hesitate to email me. You know, you may have to digest the ideas as well.
Yeah.
So if you have any questions, don't hesitate to email me, and I'll happy to respond as much as possible.
And I just wanted to highlight too that if you're a trainee, or if you're looking for some place to train or continue your training, Sathish's lab is just a great place to do research, and he's been extremely productive, and it's an exciting environment, not only in his lab, but also OMRF. It's a great place.
Yeah, we are recruiting. Thanks for mentioning it, Mike. So we are recruiting. So if you are interested, please contact me. I'll be happy to support.
All right.
All right.
Thank you once again, Sathish. This was wonderful.
Thank you, Mike. Thanks again, and hope to see you soon.
Bye-bye.
Bye.